So welcome to the Agriculture channel. Today we are going to discuss about the most important concept behind the mass selection. So in this case, first of all, this mass selection is majorly applied to self-pollinated crops. Though it was though it was also applicable to cross-pollinated crops, but it is very less used in cross-pollinated crops, but mostly used for the self-pollinated crops. So first of all, we have to understand what do you mean by the mass selection. So here you can see the mass selection majorly, that means a large number of plants of similar phenotypes are selected and their seeds are mixed together to constitute the new variety. This is the concept. So the concept is we have to select, that means what will be the original population. So here our original population will be the mixture of several lines, with non-uniform in nature. Majorly they are non-uniform in nature. Okay, and out of this mixture of several lines or mixed, we are going to select. We are going to select some. We are going to select. We are going to select with some desirable trait, or we can say phenotypic character. So by choosing some desirable phenotypic character, we will select from this original population, and what will be the selected population. So here our selected population will be somewhat uniform. Our selected population will be somewhat uniform as compared to this one. Somewhat more uniform in nature, and mostly they are homozygous in nature. Why homozygous? Because it is mostly applicable to self-pollinated crop. Because as you know that most of the self-pollinated crops are homozygous in nature. Homozygous means either capital T, if we will see either tall, capital T, or small t, we can say the dwarf. Suppose with respect to height. So the similar allele will be there. That means homozygous means similar allele will be present. Either capital T, dominant allele, or recessive allele, that is small t. So basically here what is in which case this mass selection is applied. First one is improvement of desi variety or local variety. Second one is purification of the existing pure line varieties. Okay, so let's coming to the improvement of desi variety. As you know that whatever the local variety we have, after growing this local variety year after year, so now they are going to mix with other variety. Means now it is not pure. Yes, after growing this local variety year after year, due to mixing of seeds of other variety, now it is not pure. Okay, so basically it is, we can say, the local variety are the mixture of several pure lines. First of all, what do you mean by pure line? Pure line, it is nothing but the progenies of self-pollinated crop, because these are homozygous in nature. Okay, so now the thing is that whatever the local variety we have, these are basically mixture of several pure lines. So as these are mixture of several pure lines, so in these cases there may be differ in flowering. These pure lines may be, they are different in some characteristics, either by means of flowering, or with respect to yield, or with respect to disease resistance. That means some of the pure lines will give more yield, some of the pure lines will give less yield. Some of the pure line will show disease resistance, some of the pure lines will show disease susceptible. That means now whatever the local variety is there, it is nothing but the mixture of several pure lines where both good and bad features are there, good or bad phenotypic characteristic plants are there. So now the concept is here to identify the plants which are good traits. Those plants will be selected. Those plants will be selected with our desirable phenotypic characteristics. Whereas other plants which are not desirable, they should be eliminated. They should be eliminated. So this is the thing where we are using to improve the local variety where there is a mixture of pure lines, and now we are going to select the plants with some desirable phenotypic characteristics, and now the final plant we will get, it will be more uniform as compared to the previous one. Okay, so here the original population we have seen, the original population is not uniform in nature because mixture of several pure lines are there. So some are, we can see some will give more yield, some will give less yield, some will be so disease resistance, some will so disease susceptible. So there is not uniform nature. So out of this original population, we are going to select some plants which are with some desirable phenotypic characteristics, and now my selected population will be somewhat more uniform as compared to the previous one, and as it is mostly the applicable to the self-pollinated crops, majorly it will be homozygous in nature. Okay, then next thing is that purification of the existing pure line varieties. Obviously, as we are going for cultivation of these pure line varieties, it will come across through other varieties. As you know that during the seed production program, or we are going for any cultivation program, there is a chance that mixture of seeds will be there. Suppose we are going to for rice variety, we are going to cultivate the rice variety. So rice variety, there are different types of variety, 1 2 3 different types of varieties are there. So there is a chance that in the cultivation process, and whenever we are going for to process it, whenever we are going for processing the rice seeds, in there is a chance that there will be some mixture of the seeds from B1 B2 B3, some of the seeds will get mixed together. That is why whatever the pure lines we will have, suppose B1 is a pure line, B2 is the itself it is a pure line, B3 is a pure line, but during the time of processing there is chance that there is some mixture of seeds. There is chance that from in the B1 some of the B2 will fall, or the B2 will mix to the B1 seed. So now the pure line varieties which was already in a pure form, now it will get non-uniform in nature. Okay, due to, we can say, whenever due to the mechanical mixture, we are saying that due to the mechanical mixture of seeds during the time of processing. That is why again we are going for the purification of the existing variety, pure line varieties. That is suppose B1. Okay, so now I hope you can understand all these things. So whenever we are going to procedure, let's coming to procedure. This is most important. So first of all, whenever we are going to procedure of mass selection, we have to remember that how many years? Normally it will take 6 years. Normally it will take 6 years. Out of this 6 years, the last, that is 6th year, will be used for seed multiplication program, because if the variety will be released, then we have to go for multiplying the seeds to distribute to the farmers. Okay, so now 6th year, and we need at least 3 years. We need at least 3 years for AICRP trial. We can see AICRP trial. What do you mean by AICRP trial? AICRP means All India Coordinated Research Project. All India Coordinated Research Project, what is the aim? Aim is to identify the promising variety whether is more whether the promising variety whether it is more we can say as compared to more either desirable phenotypic or other trait as compared to their agroclimatic zone check variety. That means basically AICRP, what it will do, the AICRP project, that is All India Coordinated Research Project, this AICRP present in every agroclimatic zones of our country, that is India. Every agroclimatic zones of the state and we can say state or the nation, AICRP project is going on, and the function is to whatever the variety will be released, these varieties will be checked with respect to the check variety of that local region, of that agroclimatic zones. There is a best variety of this agroclimatic zones. It will be compared with the releasing variety. If the variety which is going to release will perform better than that of the check variety, then this variety will be released to all the agroclimatic zones. Okay, or it may be released with that agroclimatic zones which is suitable for this variety, because as you know that the variety is depend upon the environment, and agroclimatic zones means different environment is there. So it doesn't mean that this variety will give better performance in every type of environment or in every type of agroclimatic zones. Hence this variety may perform better in some of the agroclimatic zones with some environmental conditions, and maybe this variety will do not perform better as compared to their local based variety due to the adverse environmental condition with respect to the variety. Okay, so the function of All India Coordinated Research Project is to identify the regions on where the released variety will perform better than that of their local, or we can say already adopted local varieties of these regions. So first, coming to the first year, let's start the procedure. First year. So in the first year, what we will do, as you know that what is the original population? I have told original population is mixture of several pure lines but which are not uniform in nature. Not uniform in nature because of the either local variety during the time of as in the local variety cultivation, we are not maintaining the agronomic we can say standards or seed certification standards. Hence there is a chance that at the time of cultivation there will be mixture of several seeds. Similarly, there should be some mechanical mixture will be there, maybe some mechanical mixture of seeds of different varieties will be there. Hence original population is not uniform in nature. So whatever the original population is there, it is not uniform in nature. Okay, and this is nothing but the mixture of pure lines, several mixture of pure lines. So what we will do, so from this variable population, variable means they are not uniform in nature. So from this variable population we will select 200 to 2000 plants. How many plants? 200 to 2000 plants we will select. We will select, suppose, based upon the, always remember, whatever the selection will be there, this shall be based upon the desirable phenotypic character. Suppose we want to select a high yielding variety, so those plants which will produce higher amount of yield, we will select these plants. So if the desirable trait is to select the disease resistant plant, that means we will select those plants whatever may be the yield will be there, we will select those plants which are disease resistant in nature. So based upon the desirable trait, okay, we will select some of the similar plant. Why similar? Because already the original population is dissimilar. Already the original population is dissimilar. Hence, we will select some of the similar plants with desirable traits. How many number of plants will be selected? Out of this, 200 to 2000 plants will be selected. Plants will be selected, and whatever the seeds are there now. Suppose I am selecting this plant, then this plant, then this plant, then this plant. Suppose whatever the plants I am going to select it, the whatever the seeds we will collect, we will composite it. That means whatever the seeds I am getting, so the seeds here I am getting from all these plants, whatever the seeds are there, they will be composited. That means they will be mixed together. Okay, the selected plants are composited. This composited means the seeds will be mixed together. Now what will happen is, it is already we are applying into a self-pollinated crops, and there is the thing is that here we are not going for progeny test, no progeny test. Hence we are going to mixing the seeds, that is we are compositing the seeds, because there will be no progeny test. Okay, so whatever the seeds we have now, the composited seeds are planted in a preliminary yield trial along with the standard check. Suppose in our Odisha region, in that, suppose here we are in Odisha. Okay, suppose in Odisha and Bhubaneswar we are going to select, we are using the mass selection variety. Okay, so in Odisha region we have some variety. Suppose it is a rice plant. Suppose a rice plant with giving, suppose it is a drought resistant. Suppose we are going for drought resistant variety, and we know that Sohavagi Dhan is a drought resistant variety. But this Sohavagi Dhan, due to the you can say year after year, this Sohavagi Dhan is now getting impure because of the mechanical mixture with the other seeds. So now our desirable trait is, what is our desirable trait? That is drought resistant. Our desirable trait is, that is the drought resistant, for example. So in this case, what we are going to do? So we are having standard check. What is the standard check? So standard check is the Sohavagi Dhan. Sohavagi Dhan is the standard check, which is the best promising variety of Odisha region. Best promising variety of Odisha region with respect to drought. So according to your desirable characteristics, you should have in that region standard check. So whatever the composite seeds will be there, we will grow these seeds. So whatever the composite seeds we have, we will grow these seeds in a preliminary yield trial. So now we are going for, this is nothing but the preliminary yield trial, and here the local standard check will be used. Okay, and now again, suppose here I have taken some standard check. Okay, this is my standard check variety, but these are the composited seed variety. Okay, so now I am comparing all these composite seeds with respect to the standard check. So if phenotype of the selected plant population is critically, in this case the phenotype of the selected plants will be critically evaluated. Now whatever the plants we are going to select, suppose I am going to select this plant, all these things. Okay, so whatever the plants, promising selections are evaluated in coordinated yield trial at several cases. So now whatever the plants will be selected, now we are going to which trial? That is AICRP trial. Now we are moving towards AICRP trial, that is All India Coordinated Research Project trial. So that means here in the second year, it is confined to the local region. local agroclimatic zones, but in the next process we are going to AICLP coordinator, that is for several locations. We are going to use this variety of several location, and in their location they are using their local best variety for the comparison purpose. Okay, so this is AICLP trial. It is mainly used for 3 years. Okay, that is from 3rd year to 5th year. And suppose this is the agroclimatic zone 1, this is agroclimatic zone 2, this is agroclimatic zone 3. Likewise, we will going to going for comparing the varieties. But agroclimatic zone, the local standard check variety is S1, this is S2, this is S3. So what is the variety we have? We suppose this is B1, B1, B1. So we have to see whether this B1 variety which is coming from this mass selection, whether it is better, whether it is better than this or better than the local standard check variety or not. So if the variety will perform better than that of the local agroclimatic zone variety which is already existing, then we are using this variety for seed production program in that agroclimatic zones. If another agroclimatic zone the variety is not performing better, then in that agroclimatic zone the variety will not be applicable to this agroclimatic zone. Okay, so once the outstanding variety is will be selected, so now it will be released as a new variety for that agroclimatic zones. Okay, so then we have to, after releasing as a new variety, we have to multiply the seeds for distribution to the farmers. So here we are going for distribution of the we are going for seed multiplication and distribution of the farmers. So this is the procedure. That means here we are, what we are having, we are having initially we are having a mixture of pure lines. Okay, you can, if we will see here in this case, what we are having, we are having initially a non-uniform variety is there. Initially here you can see a non-uniform variety is there, but here whatever the variety is there, it is more uniform in nature. Though there will be, as we are going no progeny test, hence here we are having mixture of several pure lines. As we are not going progeny test, hence the final population will be mixture of several pure lines, and but it will be more uniform as compared to the original population. Okay, so if we will go further mass selection, then the purity or the uniformity will go to an extreme point. Okay, so then I hope you can understand the procedure. Then next, coming to, if we will see what is the merits of the mass selection and what is the demerits of the mass selection. Okay, this is the thing. So let us we are discussing the merits of mass selection. So as you know that what will be the adaptability. Adaptability means whenever any adverse climatic condition will be there and adverse environmental condition will be there, so whether the plant will survive, whether the crop will survive or not survive. So here the crop will be more adaptable. There will be wide adaptability. Why? Because there is a mixture of several pure lines. Because there is a, though they are uniform in nature, but there is a mixture of several pure lines. So there is a chance that if one pure line will be disease susceptible, but other pure lines will be disease resistant. Hence your whatever the variety you have, whatever, suppose it is a rice, and you are going to cultivate the rice crop and you don't bother about the varietal characteristics. So in this case you have the mass selected variety which are having several pure lines. Okay, so in this case suppose one disease will be there, so some of the pure lines will be getting killed or it will be susceptible to this disease, but whether other pure lines, they will be so resistant characteristics, and hence this rice crop will show more adaptability as compared to the single pure line selection. As compared to single pure line, it will give more adaptability. Okay. Then next, coming to time and cost involved. As you know that in this case only how many year is required? 6 year is required. 6 year is required. So basically less time is required, less time is needed, and with respect to cost, there is no maintenance. There is no maintenance as we are going, as where there will be no progeny test and no individual plant progeny row will be there. So the cost will be also very less maintenance is there. So coming to genetic variability, yes, there is genetic variability is there, because whatever the plants are there, we are going to get that is basically homozygous in nature, but several pure lines means heterogeneous in nature. Heterogeneous in nature. Homozygous means, as you know that also. Okay, so let us understand what is homozygous and heterogeneous. Okay, so here we can say that homozygous, two things. One is homozygous. Homozygous means I have told same allele, so either dominant or recessive. So this is suppose so this is homozygous in nature, because all alleles are present in homozygous condition. Okay, so heterogeneous means genes will be different. So suppose I have two lines. This is pure line 1, this is pure line 2. So I know pure line means pure lines are the homozygous in nature. So suppose pure line 1 is TT, then AA, then BB, then CC, then PP. Suppose this is whether it is homozygous in nature. Yes, homozygous, because whatever the genes are there, 1 2 3 4 5, so 5 genes are there, and the alleles will be alleles are in homozygous condition. So this is homozygous. Suppose pure line 2 will be XX, then we can see DD, FF, then YY, then QQ. So here it is also homozygous. This is homozygous. This is also homozygous. But the gene is different. Here the gene is different. Here the genes are different. So both doesn't coincide the genes. So these, if we will mix pure line 1 plus pure line 2, then it becomes homozygous in nature but heterogeneous in nature. Okay, so if we'll see homozygous, homogeneous, if I will write homozygous and homogeneous, then it will be, suppose I will say pure line 4 pure plus pure line 5. So now pure line 4 will be, first one is homozygous will be there, both are homozygous. That means here TT, AA, suppose here RR, SS, then we can say MM. So this is homozygous. Yes or no? Homozygous. Achha. But in this case here we are going for TT, AA, RR, SS, MM. Here also homozygous. But you can see here the genes are same in both pure lines. The genes are same, though both are homozygous in condition. But here you can see dominant alleles are there. Here recessive alleles are there. But both are of same gene. Okay, so if the homozygous condition is there, but the genes will be same, then we are saying it is homozygous and homogeneous population. But in here, what we are getting here in mass selection, here in mass selection, our final result is homozygous in nature but heterogeneous in nature, means genes are different. For this, as the heterogeneous in nature, hence there will be wide adaptability. The wide adaptability because of the genes are different. Suppose this gene will be susceptible to disease, but this gene will be resistance to the disease. So the wide adaptability is because of the heterogeneous in nature, not because of the homozygous in nature. Okay. So in this case, if we'll see genetic variability, yes, genetic variability is there because the plants we are getting, it is homozygous in nature and heterogeneous in nature. Then next, coming to demanding of the breeder. So as you know that we are here we are going for mass selection. Here we are going for mass selection, no progeny test, and this is, we can say, the natural selection. This is also called as the natural selection. Here mass selection is there, natural selection is there. So the demanding of the breeder is less as compared to the pure line selection, pedigree method of selection, where the breeder should involve himself or herself completely to the selection process. But here you have to grow the seeds, then collect the seeds from the plants, and we are going for composite seed, and then go for preliminary yield trial and coordinated research trial. So demanding the breeder is somewhat less as compared to the other methods. Okay. So what is the demerit? What is the demerit? Because non-uniform nature, though original population, as compared to original population, the mass selected varieties are more uniform in nature, but still, as there is a mixture of several pure lines, so they are somewhat non-uniform in nature. And because of this non-uniform nature, they are less liked by the farmer. Because always the farmers want a variety with having similar phenotypic characteristics, similar uniform in nature. So as compared to the pure lines, selection, the mass selected varieties are less liked by the farmer, as they are non-uniform in nature. Then coming to less improvement of the variety, yes, obviously. So as there is a mixture of pure lines are there, so obviously there is a chance that we will get further mass selection and the uniformity will be increased. So there will be less improvement of the variety. So there is more, we will go for the mass selection, then more improvement will be there. Then absence of progeny test, yes, here we are not going for progeny test, but Allard has suggested to go for the progeny test for getting the whether it is homozygous in nature, really. So you have to understand. So though it is a self-pollinated crop, but there is a chance that there is, so in some cases there is cross pollination will be there. So for example, in rice, there is a chance that 3 to 4% cross pollination chances will be there. So this cross pollination will make it, we can say, heterozygous in nature. So progeny test may be there according to Allard to know whether they are homozygous in nature or not. Then coming to not commonly used, unlike pure selection, yes, obviously, because it is non-uniform in nature. Hence it is not commonly used as like in pure line selection. Pure line selection means only one pure line will be there, single best pure line will be there. So though in pure line selection the variety is released only single pure line, this is single best pure line we are achieving. Due to the, as we are going for progeny test, here we will see in the next chapter. In the pure line selection, we are going for progeny test and we are selecting the homozygous plant purely, and we are going to select the best single pure line variety. Hence it is more uniform in nature. Hence more liked by the farmers. Hence not commonly used in mass selection varieties, non-uniform nature, not commonly used. Then difficulty in identification seed certification program, yes, as non-uniform in nature, the same answer as the non-uniformity in nature. Hence it is very much difficult to identify the exact variety for the seed certification program. Then next coming to utilize the variability already present in the population. Yes, as we are going for natural selection from the original population. Here original population, whatever the variability is there, it is present naturally. We are not creating, suppose we are going for crossing variety 1 and we are going to variety 2 and we are getting variety 3. It is the crossing is there, but here in mass selection we are not going for the crossing. So whatever the variety whatever the variabilities are there in the existing population, the variability is there, we have to utilize all these variabilities. So there is no chance to create a new variability. What we need in this hybrid method, hybrid cross method. So this is not applicable. So our existing variability present in the population, we are going to utilize all these things. Okay, so this is the thing of mass selection. So I hope you have understood all the concepts very clearly, and if any doubt is there, please write in comment section. And in the next chapter we will cover the pure line selection and we will compare both mass selection and pure line selection, and you will say which one is best and which one will be applicable to what circumstances.